talked about ProDy, you now saw how it's being used. And what Jihan started yesterday, he's going to continue today. Essentially, he will show the ensemble analysis. That's what he's going to begin with. He did some of it. He did the PCA yesterday, but he will complete what he has done. And again, this type of analysis for us is, one is to make sure that what we are predicting is relevant to what has been observed. And secondly, what is new, apart from what has been observed, can we learn more and then take advantage of that? One major area of application some of you may be familiar with is when you try to bind a ligand, a drug, to a target protein. You need to take account of the conformational flexibility of the protein. However, in most cases, most docking software would work very efficiently by assuming the protein to be rigid. So this lets you generate a reasonable ensemble of conformers, which you can then use for your docking simulations. So there are many, applicabilities, but that is one which I think colleagues found very useful. Oh, that is also, for those of you who would like to see more, learn more, that was a review we wrote a couple of years ago, just summarizing many things that I have described yesterday. So when you apply to a series of proteins, usually the first mode or one of the first softest modes will provide an agreement or a correlation cosine of about 0.75 with the experimentally observed structural change. So that was, we showed that with the help of several examples. And this is, when you push you can start from both ends. You can start from this end and from that end. You can push them to get closer. But you won't be able to do the entire trajectory, but at least it gives you the direction. So that's what it is rigorously, useful for, is to give you some direction of motion. Now you've seen that. I'm going to just go very fast. This was showing several features of ProDy. You have already seen this type of Kirchhoff matrix representation. That is an aspect I briefly mentioned. We were plotting here the modes, the different modes. This is the mode index here. And there it is the level of correlation between that particular mode and the change in structure that has been observed experimentally. And usually, so this is called, the technical term for that is overlap. Overlap between experiments and theory, if you like, based on the first mode, second mode, third mode, etc. So you can see that usually the top 3, 4 modes will be approaching a cumulative correlation cosine, which is found by taking the square, those elements, summing them up. Each correlation cosine is squared first, then summed over all squared correlations, and then you take the square root to get the cumulative. So you can see that a few modes are usually sufficient for describing what is observed. So that was a, and you are now familiar with a suite of tools that I described yesterday. ProDy, of course, and Evol will be one based on sequence analysis. Jihan will show you how to use it. Druggability will be another one. That is just for setting up a system that will be done. You will then use it as input for MD simulations. But now, as I will explain, you will have a system in the presence of probe molecules representative of drug-like fragments. And then this is the visualization tool, which you have already seen. I'll spend some time talking about this membrane version, how membrane proteins embedded in a network, in a lipid bilayer, which itself is modeled as a network, are being analyzed. Just a background on that. Mechanical stiffness is another property that can be easily calculated, and we have been Carolina, another postdoc, will talk about that. We'll show you some examples in the afternoon. The major utility would be to understand this time we're really talking about a mechanical stiff, like in AFM, atomic force microscopy experiments. When you try to pull apart, let's say, two residues, what is the response? So you can relate this to single molecule measurements. And that is an extension, a hybrid model, I would say, between that combines collective modes from the GNM, ANM, and the MD trajectories to generate, lots of conformation in the conformational space. The energy landscapes, you remember that I've shown, are generated using co-MD. So that gives you a broader ensemble of all conformational space, including those that are surmounted by local energy barriers. Okay. Now, one thing that the ProDy website has is a very detailed and very well-written tutorials pages. So I would, of course, during this for 2 days here, you will have a feeling of what it does. But it is, frankly speaking, it is extremely easy to follow all the steps in the tutorial, repeat everything, and learn all the features. So it is really very well written. Same thing for the dynamics, but the ProDy, I don't remember now how many pages, but there are thousands of pages of tutorials with lots of illustrations, so you may really take advantage of that. Again, a major feature was if you have a sequence, and today I think Jihan will show that one too, you can do a blast search of the PDB, try to find where this sequence is located in which protein, or that has been structurally resolved. You can construct biomolecular assemblies. This type of structural alignment is not so easy. You can do it easily. And then determine functional motions. And you will see these things. You have already seen some. And the major two models behind all the theory are the Gaussian network model and an isotropic network model, which are explained in detail in that component of the tutorial, elastic network models. In the second part of this workshop, you're going to see how you're going to become more familiar with MD trajectories. The ProDy interface can be used for analyzing MD trajectories as well. And we found that many people are taking advantage of that particular feature. And then from the MD trajectories, again, you can extract the principal variations, the principal changes in structures that are observed. Okay. Now I will talk about ANM. I know that many of you are membrane. For lack of a better word, we call it membrane, but now it is really very difficult to pronounce it. Now we have the idea is for a membrane protein, we don't take into account in protein, GNM, ANM, the environment. So we assume that water molecules, for example, will be accommodating somehow the structural changes of the protein. So the protein rules, in a sense, and water molecules will be just rearranging themselves. But then this assumption may become a little bit too strong when it comes to lipid molecules, which are not as flexible and small as the water molecules. And we have realized in a couple of applications that we obtain better results if we take account of the fact that lateral motions, radial motions against the membrane will be resisted by a higher force compared to axial motions. So if we are talking about the sliding of the protein, during which it experiences mainly shear forces, that is easier. But if you are talking about some deformations that push against the membrane, they are more difficult. And we observe that in many cases, we are predicting, overestimating the motions against the membrane. We need somehow to restrain them. So that was one motivation. Yes.
Can you change the type of membrane that you would have there?
Am I?
Type of membrane.
I'm sorry, I didn't hear.
Can you change the type of the membrane? And
No, we don't have that. That's a very, very good point. Actually, we need, we just started, to implement, and so if you have seen its utility, I will show you an example. But this is something others also asked. Because the composition of the lipid bilayer may be important, then we may need different parameters. We haven't gotten there yet. So you can use either ProDy or dynamics for that. The same theory has been implemented. I'm sure some of you will like it better to use dynamics, but ProDy has more features in general. And that is the example I think Honchan showed yesterday for a membrane protein, the gamma secretase. So these are the two modes he mentioned, one of them. So this is the gamma secretase, this is the transmembrane region, and then there's this extracellular part. The two motions, which you can view either as static images or like movies, essentially one is the bending, and the other is a torsion motion. It's hard to see it from that direction, but I think you can see it better from the top, how the top portion rotates in an opposite direction to the. So that is, I think this is the example that is given in the dynamics interface, to walk you through about all the steps how to do, and that's a useful one. I will talk about and that was another example which I gave yesterday actually, the AMPA receptor. Remember I mentioned the fact that by cross-linking a pair of residues here, we are able to verify that indeed they can come into close proximity, and if we block that, there is a consequence. That's something I mentioned, and today James Krieger will go into more details on that. Now, the example that motivated us in the first place was a glutamate transporter, and I would like to spend some time on that one now. So glutamate transporter is a trimer. This is the side view. So this is the part embedded in the membrane. So you have a few extracellular loops and a few intracellular regions here. And from top, the same structure, this is the way it looks. So there are 1, 2, 3 monomers, 3 subunits. The blue region is like a scaffold that holds the trimers together, intertwined. And then you have the red-orange regions, which is the transport core for binding the glutamate. Their role is to clear excess glutamate from the synapse. So this is another view. Originally, when the structure was determined, they defined, based on structure, they defined 2 substructures. One was this substructure here, which we can see from the other perspective, composed of 2 helical hairpins, HP1 and HP2, and 2 transmembrane helices, TM7 and TM8. So this is the transport core. And the remaining regions, you have plenty of other transmembrane helices, would be like the trimerization support, if you like, for all of them. So this is the way they at least characterize the structure composed of something required for the support and stability, and the other one required for the function, okay? Transport function. So this is a closer view to it, one of the subunits. So all the activity takes place near this HP1 and HP2 loops. That's where a drug binds, or the glutamate binds. This is a secondary transporter in the sense that the transport of glutamate requires co-transport of sodium ions because it takes place against its a potential increase. Sodium ions, on the contrary, they go downhill both chemically, electrochemically, let's say, in terms of their chemical and concentration gradients. So it needs to be co-transported with sodium ions. So this is the region where the sodium and glutamate molecules, sodium ion and glutamate molecule, bind, okay? All right, and this is all the most general mechanism for that has been alternating access, the way nature copes with that. What does it do? There is, in humans, these are called excitatory amino acid transporters, EAAT, okay? And so the excitatory amino acid and the sodium ions and even a proton that's being transferred first comes from the exterior. The exterior would be, for example, the synapse. And while it is in the outward facing state, so this part closes down, and then there is a release of the cargo when it is inward facing, and then it goes back to outward facing. So alternating access means outward facing and then inward facing. So this type of motion, okay? So that's the general model for transporters, but you'll see that in this case it's a little bit different. Now we do our ProDy analysis. You can do that. Anyone can generate. And we saw that these are the first two modes. This one is actually asymmetric movement of the subunits. And there are two such modes. It's doubly degenerate for those of you who want more details. That's the third mode in a strict sense, which is fully symmetric. So this is like an opening, closing of the top portion. And this is an asymmetric opening. Now, when we generated that, we thought there is a problem. So what we are generating is this type of opening, right? But we're not doing the inward facing part. There is no opening in the back. So we just have about half of the motion, if you like. Here. Now, this is the side view of this symmetric movement, or the top views again. Exactly the same logic as the AMPA receptor. Susan Amara, my collaborator, currently the scientific director of NIMH, she did experiments to cross-link residues located here, and we have seen exactly the same thing. Originally, those residues located by more than 40 angstrom apart were able to come together to form crosslinks, and this effect was reversible. So we were able to see how cross-linking affected the glutamate uptake. So that is, we can predict this type of, this motion indeed takes place, that's true, but it is incomplete. So then we have, here are the two actually originally the structure that was resolved was the outward facing, and a couple of years later the inward facing also has been resolved. So from top they look very, very similar, as you can see. From the bottom, from the side, you can see that this is the outward facing, and the inward facing would have some regions more exposed. So this is the way it is. Now, the idea was, if we examine more closely what's happening, and I think this is really a fascinating example. The outward facing single subunit here in the original state. Don't worry about inhibitor, but it's still outward facing, whether it is inhibitor bound or substrate bound here. When it transitions into an inward facing state, that region goes all the way. You remember that was the transport core. It goes all the way toward the cytoplasmic region. So here, that's a schematic representation. You have a translation of this core by about 15 angstrom, which is really something very extraordinary to my view. But there is more than that. So that was the way they described in those original papers. But there is more than that. If you analyze carefully, it's not only the transport core, but also there is another helix here and another helix here that are also being displaced at the same time. So we have those TM3 and TM6 here, which are also displaced. So the original separation of the domains, TM1 through 6 is one domain, and TM7, 8 together with the two hairpins is another domain, doesn't work because part of the first domain actually is more engaged, moves together with the second, okay? TM3 and TM6. So there were this type of interesting observations from experiments. And this is also showing the motion. So this is the way it moves, right? When it transitions. So what it does, essentially, it is really beautiful. In the outward-facing state, it takes its cargo, the glutamate sodium ions, and then it just moves down and then releases them. So it moves up and down. It's like an elevator. This has been called an elevator motion. All right. So now the question was. The
transport occurs on a single subunit?
Each subunit transports a glutamate, yeah, each of them. Now, there were also some arguments whether the subunits function independently. But now we know that there are those cooperative movements. They have to work together, but each of them, yeah. So the question was, we would like to see whether, you remember this diagram from yesterday? This is one substate, outward-facing substate, let's say. And suppose this is the inward-facing. And can we really predict that we are going from here to here with the help of one dominant soft mode? Okay, that was the question. And we already knew that it would not work because we have this type of motion. We don't have this vertical movement you've seen already, right? There is something missing. So then I am asking the question which I already answered. So we would like to see the following. Here is the outward facing, here is the inward facing conformation, okay? Now I'm taking the difference between them, subtract. The difference is what we called yesterday the deformation vector or difference. We can call it a difference vector. So you just, after optimal superposition, and that would be based on the membrane position too, because you don't want to align them, actually they are shifted, but you align them with respect to the membrane. This deformation vector that we generate is just, you take every single atom coordinate, in our case alpha carbon coordinate, x, y, z, and you take the difference, you generate this 3-dimensional difference vector. So the question is again, once we have calculated this dn, and we did our analysis, so we can do our A&M analysis either for the outward facing or for the inward facing. We can try to see both ways, right? We want to see whether there is a correlation cosine which is high enough to support the predictions, right? And as I mentioned earlier, when you take the correlation cosine, square them up, sum over all modes, and then take the square root of the summation, by definition, this is equal to 1. So the modes, the spectrum of modes, provides to you like a complete set of all accessible motions one way or another. But what matters is that whether those observed experimentally are the easiest motions, are the most probable motions, okay? Whether they are among the soft modes. So Tim did that actually in almost 5 years ago now. This original study, he came up with the idea when he was a postdoc in my lab. Now he's a faculty member. And he is, the idea is exactly what this is a slide from yesterday, what I've shown for the Groyal case. We have all the modes, and we plot the correlation cosine between the modes and the difference vector, right? And in that case, I very happily showed you that mode 1 gives a correlation of more than 0.8. Now, in this case, when we do the analysis, when we do the analysis without the membrane, it looks like that's what we get. Now, one of the curves is for this one. We start from that, and you would like to reach the other one. We need at least 30 modes to approach 0.6. And now the other one, the other curve is even from that one. So of course, eventually, if we take all the modes, we're going to approach 1. But that's not the way we would like to see things. There is not a single. So here, I've been plotting the cumulative directly. Okay, it's not. You see, the first 10 modes, they don't do much here. So we were very disappointed. And then with membrane, that's the difference. When Tim took into account the membrane, remember there were 2 asymmetric motions. They are here. But then the third mode that was fully symmetric completely changed, such that now we reached, with the help of this third mode, a correlation of 0.6. And I'll show you what the motions are. Here is Tim again. So let me explain what we're doing again. We start from this conformation. In the presence of the membrane, now we are generating a new conformation. Or we start from this in the presence of the membrane, we generate a new one, which is really an intermediate between them. And it provides a good description of what is happening, which I'm going to illustrate with the help of a movie. So we came to the conclusion that the membrane is actually assisting the protein in taking this type of movements. This is not the property, the character of the protein alone, but the protein embedded in the membrane, which is moving now in concert with the membrane. Just as a remark, normally we have 3 N modes, right? For N is the total number of residues in the trimer. And normally when you do the cumulative overlap, each mode contributes by that amount. If everything was random, all the motions are totally random, there is no any meaning to them, that would be the contribution of each mode. Now we're talking about a mode that contributes by 0.86. So that shows how much actually this is the, if you like, the enrichment or the improvement compared to random that you can judge. Okay, here's what he did, what Tim did. This is called, and this is again, not only our lab, but our other labs use similar approaches, not for membrane proteins, but for system and environment in general. So this is the protein, viewed from top again. And now he embedded that in a membrane environment, where the membrane molecules are also represented by beads and springs. Now, in this case, this is just a schematic, just to illustrate. But the idea is now you build a big Hessian matrix. The system is the protein, and the environment is the membrane. So that part of the Hessian is what we used to use only in the absence of the membrane. And that is, this part is only environment, the interaction between the lipid molecules. We can elaborate on that, but we don't have to. And this is the cross correlations, basically at the interfacial residues. Some of them will be located here. Now we have a new expanded Hessian that comprises both the system and the environment. And in this case, you have also, it is possible, and I'm not going to do the derivation, but it's very, it's straightforward to show that this part of the membrane may be approximated, this part in terms of an operation involving the other parts, such that this approximate Hessian may be utilized then to understand what's the behavior of the protein now in the presence of the membrane. So of course, if this Hessian is not so big, in many cases you can directly use it, obtain its eigenvalues, eigenvectors, but that's an efficient way of doing if you have a really huge system. And the command in ProDy to generate this type of reduced model for the system in the presence of an environment is that one. I'm not sure if in the afternoon you will show it. So that is, that's the model. Now this is, we call that the explicit model, where you have explicitly nodes representing the membrane, right? This is the explicit. And then a team also came with an implicit model. And in the tutorial, you can see the explicit and implicit models. Now, the implicit model, it's even smarter, I think. The only thing he did, he introduced a new parameter. A new parameter to change the force constants. Let me see. Maybe it is a little bit too complicated here. But what he defined, you know, you think we have been using a uniform force constant, remember, for everything. Now we're going to use two different force constants. One will be for radial motions, and the other will be for axial motions. So you can define your x, y, and z. Now, for the x and y, which is radial directions, you're going to have the same force constant. For z, you're going to have another force constant, which will be smaller because this z direction is easier, remember? So the new parameter, the single parameter, is the ratio of the force constants between the radial and axial directions, essentially. So now he defined gamma x, gamma y, gamma z, but y and z are the same, and what matters, again, is actually the ratio, this thing. So the net effect is that you do still the A&M analysis here, but now you have different force constants for those movements, those against the membrane versus sliding in parallel with the membrane. And that option is also, you can use that command in ProDy for that particular option. And now here's what happens. I think this movie is with the implicit approximation, but it was really good for us to see that qualitatively we obtain the same results practically either way. So that's the net effect of the membrane. So here, this is what we obtain in the absence of membrane. Okay. This is the motion, remember the third motion, and now we are rotating. This is what we are observing. You can see there's a slight movement, vertical movement, but not much, okay? Now, let's see what happens in the presence of membrane. Let's see if you're gonna see the difference. First of all, these movements are really very suppressed. Now, if we look from the side, look at that. We have the elevator motion now. And this is the, you know, you obtain this solution as the third mode that provides a correlation cosine of 0.6 with experiments. That gives us really a lot of, you know, support for the idea of adapting, taking into consideration the membrane in the first place, and the fact that those approximate models could be used. So you want to see again? Let's see both of them while I'm answering Steve's question. Okay. Unfortunately, they are not. All right. So you can compare now. They are similar. but that one really undergoes larger up and down movements. You had a question?
So how do you tune the magnitude of the force constant?
We don't know. We don't know.
Now, you must put in a value for it.
That's true, and we always. So what we have been doing, whenever there is an X-ray structure, we have been adjusting the force constant, a single parameter if you like, to get the absolute value of fluctuations. Now we've done a systematic analysis of all PDB structures several years ago, and it turns out that a generic number is, for proteins in solution, is 1 kcal per mole per angstrom square. So it's a little soft potential. One. So something around 1, but that is, you can, if you don't know anything, you can adopt that value. Now, in our calculations in ProDy, in dynamics, we usually, because we are comparing with the B factors, we report also what has been the force constant that has been substituted. And usually it will be around 1 kcal per mole per angstrom square. The thing is that there is no unique force constant in nature. What I was trying to explain yesterday, remember I showed that the ensemble of structures that we are observing are all along the same mode, different extents of opening, for example, of the same enzyme. So, you know, there is no need to define a single force constant and say that, okay, here is the next conformation, because actually, depending, for example, on the bound ligand or on the which stage in the cellular function it is, it may, it has all this freedom to continuously move along that direction. So we're not so concerned about the precise value. We want to know the mechanism. That's the bottom line. Now, here again, one thing we try to pay attention is we don't want to deform the molecule beyond what is physically reasonable. For example, when we start to see that bonds, alpha-alpha, C-alpha, it's supposed to be 3.81 angstrom. If now this plus or minus, let's say 0.2 angstrom. If this is exceeding these reasonable limits, now we need to be careful. Maybe we are exaggerating the motion, or we're pushing, you know, we're doing something physically unrealistic. And that's why actually in dynamics, the most recent version that we published, we have an extra term, if you like, to control, to make sure that the bond lengths, bond angles are within reasonable physical limits. So we try to pay attention to that. But otherwise, what we've learned is that there is not a single size of deformation. It is a continuous ensemble. But that's a very good point. You know, we have been thinking about what is the value. But now we came to the conclusion after 2 decades that we don't need a single value. And the values that I said, like 1 kcal per mole per angstrom square from X-ray structures, they may not, in the X-ray environment, the fluctuations are small. When we did also the same thing with NMR structures, we have always seen that the structural variability is much larger. And the GNM and ANM, they have always given better agreement with NMR data, because now you really have more motions compared to X-ray.
So the Z force constant is 1/2 of the...
Which constant?
The Z, the...
Okay. Yeah, I don't remember that. You may want to check, but it could be about something 2 to 5. I don't remember now. Yeah. Okay. Oh, remember also I mentioned this domain separation thing. Now, when we do our GNM analysis and we look at this mode, you remember that mode that's important, the dominant mode. Now, this is the way, if you use only a single mode, you get a very clean cross-correlation map. Because now you're literally, a single mode is a single direction in this n-dimensional space, right? But it is a single, now either a given residue moves along that single multidimensional direction or not. So that's why you get really very clean results when you plot the single mode in GNM. Okay? So in this case, for some reason, we start from here to here, 0, 1 to 400. That's what each subunit is in the transporter. So the red region here means this part is moving concertedly, then this is anti-correlated. So this is transmembrane helix 3 is anti-correlated with the transmembrane helices 1 and 2. So 3 is anti-correlated with 1 and 2. And then 4, 5 are correlated with 1 and 2. Okay? You have the complete map from here. Now what you can see from here is that actually 3 and 6 and the loop next to 6 are moving together with 7 and 8. That's what you can see. So the original separation of domains based on structure, which turn out to be not true when you observe the invert, 5 years later they generated the second structure. Actually, we could have simply looked at this third mode and see already that that domain separation was not correct. I don't know if this is a very powerful tool for defining domain separations based on dynamics. Looking at structure alone may not tell you the truth. You may be misled by some, but the way everything is being wired defines actually, because it is being wired like it's a network, right? So this type of diagram, if you concentrate on the dominant mode alone, a single mode, it gives you a very clear description of the different domains. So essentially, we have the scaffold, what I call the scaffold, originally said to be from TM1 to TM6, only involves those 4 helices. And the transport core now is the old transport core plus TM3, TM6. Now they corrected that in the literature. For several years, that was a wrong description.
Sorry.
Yes.
So this was calculated by GNM, right?
This was GNM, yeah.
GNM.
Yeah. But you can calculate by A&M too. I like GNM better whenever I can do GNM because it is simpler. And it has less, in the A&M you have the extra, when you bring the 3 n dimensions, x, y, z components, unless you are interested in directional effects, you bring an extra source of error, let's say, because now you split into 3 dimensions. If you are interested in the magnitude of a motion, in a cross correlation which you can generate by GNM, I would rather use GNM. But in principle, they should be equivalent. You should, you should be able to trace that the same mean square fluctuation is being predicted by both. But GNM is more accurate. It is my take. I would recommend to use GNM whenever GNM gives you the result. So that is the last slide I would like to show you here. This is the membrane protein moving, again, the AMPA receptor moving together with the membrane. So there is, whatever is happening is being accompanied by the membrane. These were also some studies that from Hong-Chan he did recently, not yet published actually. But at least without membrane, that's what it does. But with the membrane, some of the motions may be split into others together with the membrane. This is a totally new mode, and actually it's nothing else than the membrane movement itself, or how the different movements, there was a very broad fluctuation here, which is suppressed in the presence of the membrane. So you could see what is the effect of membrane in general.
So why do you show the membrane being deformed?
I'm sorry?
Showing the membrane being deformed.
Why? What's the question?
Why is the membrane deformed in mode?
Well, it is, the membrane also undergoes undulations and rearrangements, so that's what it's showing.
So that mode causes curvature in the membrane?
I would expect so, definitely, definitely. That's exactly what we're using now to explain, some invaginations formed or maybe even initiation of endocytosis. If the structural change induces negative or positive curvature in the membrane, that may be assisting in initiating some events. Definitely, yeah.
Sorry.
Yes.
So this is regarding 2 slides before this.
2 slides before.
This one. So you're saying that you could have kind of predict from the third mode that these domains
Yeah.
are working together, right?
Yeah.
So that is really fantastic.
Uh,
But how could you do that before having experimental data?
That's a very good point. Actually, you can do that for, we haven't shown that, I think, for the mode 1 and 2. I'm pretty sure, very, very certain, that if we do for mode 1 and 2, we're going to see the same thing, probably.
Okay.
So
this is not something that happened only in mode 3.
no, I think because, again, we can check that. But if there is such a trend, if mode 3 is dominant, you saw by how far it dominates. If you take all of them, all of the modes in the first place, you will see still the trend, but it's not going to be like black and white here. You will see some gray regions, but you will see the same trend if you take all modes in the first place. Now, if you take the first 3 in this case, remember how the first 2 were contributing so little. So suppose you don't know experiments at all, but now you have your mode spectrum. You have your mode spectrum, and you don't know that the first 2 are contributing little. So I don't know. In principle, I would expect to see the same pattern by summing up all modes. The reason I'm showing that particular mode is because it's very clear, very clean in a sense. But you can see. Yes, James? It does. Yeah, you can. That's a good point. But you can find, James. There. Now, the GNM gives you the displacement R square, right? ANM gives you the XYZ components. But you can move from ANM to the GNM. You can compare. You would see what would be the counterpart. You could associate them. Okay? So this is, I'll skip that one. And there are many other applications. We will move to other applications. One of them, as I mentioned, was the this AFM application to AFM. So I will just move to that one, the mechanical stiffness. And this is based on, again, a paper published several years ago. So the idea is, the idea is the following. I don't know if you can see. This was a GFP, green fluorescent protein, for which a group in Germany, they did experiments, and they have been pulling the protein at various pairs of residues. Okay, so usually in AFM they pull at the 2 ends, C and termini, but in this case they were just selecting pairs of residues, and depending on the selected residue, there is a different response. So what happens physically is the following: if a given extension is being accommodated by a soft mode, it's going to be easy to do that type of extension. But if you require really, if this deformation is only accommodated by a high frequency mode, higher mode, the response is going to be stiffer, okay? So that is the logic. And we wanted to see how much each mode contributes to the deformation of a given pair of residues. Those that are being deformed with the help of slow modes will be easy. Easier than others. So that was the logic. And So that is what I have explained in words. Now what we've done is the following. Now suppose I take this, this particular direction, okay? This particular direction is now, which has been experimentally tested. These two residue pairs. And I evaluated how much different modes contribute to that deformation. So you have many modes that contribute, but in some cases one contributes more than the other. Or that, for example, here you can't see, but there is this first mode that makes a major contribution, and the others are smaller. You can see even the scale is, let's say, 0.5 here. These are all much lower. So that motion then, because it is enabled by a soft mode, is going to be easy. Okay? And they have measured oh, okay, there's another mode here. Sorry, that one is almost 0.8. So that's going to be the easiest in this case, because it is really practically enabled by this softest mode, and others contribute just a little bit to it. So now based on this type of correlations, it is possible we were able, let me see. Oh, I'm sorry, I don't have the diagram, but we were able to show exactly how those different measurements, those different effective force constants could be calculated just by taking a weighted average of all those mode contributions. Each mode has itself a certain frequency, as you know. So by evaluating this type of distributions, you could calculate an effective force constant for this deformation. So remember, the eigenvalue or the frequency of each mode is the effective force constant, is the force constant for that mode. So if you have a distribution of modes with different weights, you can calculate the average effective force constant. So that's what we've done. And we were able to reproduce experimental data, not only for this protein, but also for many other examples. Now what currently ProDy allows you to calculate is this type of map, residue. And in newtons per meter, you would have, or you can change to piconewtons per picometer, nanonewtons per nanometer. You would have the force constant associated with deforming any pair of residues. Yes? Sure.
I'm just not sure why they're doing these studies for definitely or
I'm sorry, Tammy.
I'm just not sure why they're doing these studies, like stretching out the various amino acids. Is that how it would behave when it's
Yeah, yeah. This is one way of characterizing the mechanics of the protein. So this is a well-established method, AFM, atomic force microscopy. You know, this type of usually in most simulations, people they examine what is the unfolding mechanism, for example. So they would see, for example, one of the helices or beta strands will unravel first, and then there will be successive events. And the unfolding mechanism sometimes provides information on the folding mechanism. That's one of the motivations. But in general, to characterize a protein, a molecule, you know, I'm not going to defend why AFM experiments are done, but to me, it's another piece of information on the mechanics. All right, so this is the map that you can get. So for every single pair of molecules, now you know what is the resistance to deformation. You have those numbers. And this was the theory behind it. Maybe I don't want to go into the theory right now in the interest of saving time. However, the whole idea is to calculate an average force constant based on the contribution of different modes to the observed pair deformation. So the next topic would be to move to sequence analysis. And I wonder if this is a good time to give a break, or should I continue? How do you feel? Do you need a break, or should I You need a break. Okay, let's give a short break. In the meantime, you can ask questions if you have any, and then we will continue. Let's take 10 minutes, okay?